So this is your standard eukaryotic cell. With the prokaryotic cell, it was pretty easy for me to show you a typical prokaryotic cell because most prokaryotic cells contain very standard components, and they're all fairly, well, most are fairly similar. Eukaryotic cells are much more varied in their shape and are often much more specialized in their function. Prokaryotic cells are all single-celled organisms, so they all have to have all of the things necessary to live on their own. Eukaryotic cells are often parts of, well, like you guys, multicellular organisms. And there are actually not a whole lot of cells in your body that could exist well by themselves. Most of them can't necessarily do all of the things that they need to stay alive by themselves. They have to work in contact with the other cells in your body, or they have to be treated very gently. Like we can get them to grow outside of your body in like a cell culture environment. But speaking as somebody who's grown bacterial cells and who has done, tissue cell cultures, it's way more difficult to get eukaryotic tissue samples, like from a multicellular organism, to grow than it is to get bacteria to grow. Bacteria, just throw them on the plate, toss them in the thing, and they grow. They pretty much take care of themselves. With eukaryotic cells, if they're not in the entire organism, you have to do a lot to help them. You have to feed them special stuff because there are certain chemical reactions that other parts of the body are usually taking care of. You have to have a way to remove the waste because they don't have a way to remove their own waste. They assume your liver is going to do that. You have to keep them under like the exactly proper pH because they don't regulate the pH quite as well. And they're a lot more picky to work with. So this image that you see right here is what we would call, I guess, a typical animal cell. But there are actually very few animal cells that are necessarily going to look like this. This is a generic rather than a specific one. And not all animal cells are necessarily going to have all of these components, though they'll probably have most of the ones that we're going to talk about today, and they might have a few extra special stuff just to deal with what they deal with. Now, the eukaryotic cells are much larger, much more complicated, and the big difference is that they have internal membrane-bound organelles, most especially the nucleus. We're going to go through, we're going to talk about each of these different organelles and their functions. Now, with bacterial cells, with prokaryotic cells, we spent like this much of the time on the stuff inside the cell, and then this much of the time on all of the stuff on the surface of the cell, because with bacteria, most of the interesting stuff is going on the surface. With eukaryotic cells, it's just the opposite. The surface, kind of boring, at least relatively speaking, but the insides get really complicated. So the big difference, what makes this a eukaryote rather than a prokaryote, is the nucleus. And the nucleus is the site that contains DNA. Who remembers what is the equivalent structure in a prokaryote? Yes? The nucleoid, exactly. The nucleoid is the region in a prokaryotic cell where the DNA is contained. But the nucleus actually does more than just contain the DNA of a eukaryote. If all it did was contain the DNA, it would be a relatively simple structure. But it does that and more. This is the site of gene expression. It regulates in part which genes are going to be expressed and which genes are going to be turned off by controlling what has access to the DNA and what doesn't. Only certain chemicals can get in through the nucleus, the nuclear envelope, that double membrane structure. And if chemicals can't get in, then they can't turn on certain genes. So entrance and exit is very carefully regulated in the nucleus. by these nuclear pore structures. There are these very large, complicated regulatory gateways that control what can go in and what can go out. The nucleus contains the chromatin. The chromatin, so we don't really talk about chromatin in prokaryotes all that often. Chromatin is DNA plus protein. And if you look at it just in terms of how much DNA and how much protein there is, it's about 50-50. It's about 50% DNA and 50% protein. And this is actually part of why it was so difficult in early biology for people to realize that DNA was actually what contained the genetic information. For a long time, people thought that it was the protein that contained the good information and that the DNA was just a scaffold. Now we know it's mostly the other way around. The DNA contains the genetic information, and about half of the protein that's there is histones. Hopefully you guys have heard about histones. We'll be talking about them a little bit later on, but they don't really have a whole lot to do with microbiology. But those are the structural proteins that the DNA is wrapped around. Kind of like, this is sort of like the after Christmas season, right? So you know when you take your Christmas lights down and you bundle them up, some people just sort of throw them in a box, and what happens next year? You take them out, and it's like, up by one cord, lights just sitting there all tangled. Smart people like take the lights and they're going to wrap them around things, and they're like, just wrap them around this, wrap them around that, wrap them around that, keep it all nice and organized. That's the role of histones, because you've got enough DNA in one cell, one single cell in your body. The DNA in that cell would go from the tip of your foot to probably the top of the second story of this building. One cell. There's a lot of Christmas lights to get tangled up. So it has to be kept very organized. The rest of the protein that's there is controlling which genes are turned on and which genes are turned off. The information is in the genetic material, but using it is the job of the proteins. Now, the nucleus has a specialized structure called the nucleolus. In eukaryotes, when you turn genes on, we're going to talk about this more when we talk about genetics, but you go from DNA to RNA to protein. Hopefully everybody is generally familiar with this. In prokaryotes, it's very simple. It's DNA, RNA, protein. In eukaryotes, it goes DNA to RNA, and then there's a bunch of stuff that happens to that RNA. The RNA has to be trimmed and groomed, and it has a few little things put onto it, and then it has to have some decorations put on the back and the front, and it has to have its introns cut out. All of this is called processing, RNA processing, and that happens in the nucleolus. And that has to happen before the RNA can be exported through the pores. Any questions about this? Good. Moving out from the nucleus, the next structure that we encounter is the endoplasmic reticulum. The endoplasmic reticulum is actually contiguous with the nuclear envelope, by which I mean if you were to take, let's say, a green fluorescently labeled fatty acid membrane lipid and stick it right here on the edge of the endoplasmic reticulum, and then you were to wait a while. It'll take a while for this to happen, but eventually you would find that on the nucleus itself. So the membrane from the outer envelope of the nucleus to the endoplasmic reticulum, technically it's the same membrane. They're structured a little bit differently, but one molecule can go from one to the other. There are two parts to the endoplasmic reticulum. Again, they are contiguous, but they look different. and they have different functions. The first and probably most well known is the rough endoplasmic reticulum, or rough ER. It's called the rough ER because it has, like if you look at it under a microscope, it looks rough. And the reason why it looks rough is all of these ribosomes found to it. This is the site, it's not the only site of protein production, but all of the proteins that don't go to the basic cytoplasm are made in the ER. If it's going to go exported outside of the cell, if it's going to end up in any of the membranes of the organelles or the outer membrane of the cell, it's made here. If it's going to go to the Golgi body or any of the other future organelles that we're going to talk about, it gets made there. So actually most of the protein in the cell is made in the ER. Not all of it, but most of it. And it's made on the surface of the ER. So these ribosomes bind to the surface. That's the reason why the endoplasmic reticulum has this weird shape. It looks basically like a bunch of pancakes kind of stacked on each other. This increases the number of folds. This is something that you're going to see a lot of in microbiology and in physiology. Whenever you see this sort of motif, you've got like lots of wrinkles and lots of stacks on top of each other. The purpose of that is to get the most amount of surface area in the smallest amount of volume. That means you're dealing with a process that happens on the surface of something. And it is indeed the surface of the rough endoplasmic reticulum that matters here. The insides, things float around in the inside until they break off and they go somewhere else. The proteins get made on the surface. The smooth ER has a slightly different shape. It doesn't look rough because it doesn't have those ribosomes on it. And instead of looking like kind of a bunch of pancakes stacked on each other, it looks a little bit more like, I don't know, like a set of interconnected, like really complicated caverns. You can see here where they're like much more narrowy and branching. The smooth endoplasmic reticulum is where membrane is born. Actually, all of the membrane in the cell originally comes from the smooth endoplasmic reticulum. That's where membrane lipids are made. In addition, this is also where a lot of detoxification takes place. There's a couple of different types of detoxification that go on in the cell. This isn't a physiology course. I'm not going to distinguish them for you. But one of the places where toxins and harmful substances are broken down is in the smooth ER. Usually that's going to be hydrophobic or fatty acid or steroid-based toxins. Any questions about this? So moving out from the ER, let's say you're a protein, okay? So the ribosome assembles on the ER, the protein gets made, and it's stuck in those folds in the ER. Well, what happens is a bunch of protein that all got made at the same time gathers together, and it goes to the tip of one of the ER's little folds, and that tip will break off into a little ball. That ball is called a vesicle, and it moves from the ER and floats over to the Golgi. The Golgi is another sort of pancake stack looking thing. But it's not directly connected to the nucleus, and it's not directly connected to the ER. So the only way you get here is by floating here, usually in one of these balls. The Golgi is central shipping and receiving. Okay? Much like, I don't know if any of you have ever worked like in the back of a big department store or in a warehouse or something like that, where you get a bunch of crap that comes in, And then it has to be labeled and sorted so that, it all comes in kind of a random order, and it has to leave in an organized order. That's basically what's going on here. It's a bunch of proteins. All the proteins that get made by the ER come in on this side. This is called the cis side, which means, the close side. It's the side that faces the ER. And they arrive here and they spill in to the Golgi. Once they're in the Golgi, there are little codes on each of the proteins. They actually all have a little tag, which is a part of their amino acid sequence. They aren't mature until that tag gets cut off. So that tag has to get cut off, and that means that they have to get relabeled. And it's the Golgi that relabels them. It reads this tag, it decodes it, it figures out what type of protein they are and where they need to go, and then it's going to put a barcode on them. Okay? This barcode is usually going to take the form of what's called glycosylation. What does glyc mean? Sugar. Basically, you're going to attach sugar molecules to the surface of the proteins that come in. And these sugar molecules are the barcode that tells the cell which proteins go to which organelles, where it should end up. And what happens is that the proteins kind of float through this maze in here, and every so often they get shipped to the back and they have to float through again until they all get the labels that they need. Then you'll get a whole bunch of proteins that all have the same label on them. They all have the same barcodes. So they're all going to the same place. And they'll gather in one of these ends. So right here, this one might be a bunch of proteins that are headed to the lysosome, which we'll talk about in a second. These might be a set of proteins that are actually going to be exported from the cell. So those are extracellular tagged proteins. Maybe this one over here is all stuff that's going to the mitochondria. The point is that once you get to the other side, this is called the trans side, which means the other side, the far side. Once you get to the trans side, everything that's labeled the same gets gathered together, and then it breaks off into another vesicle that gets shipped to its proper destination. So lysosomes, peroxisomes, and vesicles. These all look pretty much the same under a microscope. They're going to look like little sphery things. Little kind of amorphous spheres. Some would be a little bit bigger, some would be a little bit smaller. Hard to tell the difference. They're membrane-bound compartments with a single membrane. They're usually going to be spherical because they're going to contain things. Whenever you see something that's spherical as opposed to wrinkly, that means that it has the least amount of surface area containing the greatest amount of volume. And that means that you've got a process that's going on the inside of something. Lysosomes are sort of like the stomach of the cell. Just like your stomach digests food for you, the lysosome digests organic material for the cell. And just like your stomach has to be segregated away from you, like if you get a wound in your stomach and the stomach acid spills out, it's actually very, very difficult for that wound to heal because it's got stomach acid pouring out of it. And it tends to ulcerate and just get worse and worse and worse over time. The same way, the stuff inside of a lysosome is actually toxic to the rest of the cell. It has to be isolated away in this little circular thingy, because if it was to spill out into the rest of the cell, it would begin digesting the rest of the cell. So if you have a cell that eats another cell, or it eats some organic material, not like food is in sugar, not food that you're going to digest directly, but something that needs to be broken down before it can be completely used in an energy process, the lysosome is where it's going to head. Peroxisomes contain reactive oxygen species. And it turns out that actually, we'll get into this a little bit more next week, oxygen's poison. And it kind of sucks because you can't hold your breath that long without it. But oxygen's actually really, really dangerous. And a lot of what your cells do and spend energy on is protecting you from all of the damage that oxygen is constantly doing to you. Peroxisomes are where these dangerous reactive oxygen species end up. The reason why oxygen is dangerous is because it likes to break chemical bonds and it likes to steal electrons from things. So in this peroxisome, you're going to digest things that don't digest organically very well. Lipids and fatty acids can get digested in the peroxisome, and other harmful toxic molecules that are hard to break down will be shunted into the peroxisome. The oxygen will go to work on it and tear it apart. This is also going to be a place where if you create any dangerous reactive oxygen species, they're going to also end up in the peroxisome. So lysosomes and peroxisomes are both to aid in breaking things down. Vesicles are like cars, right? They're temporary transport or storage mechanisms. Whenever you need to move a bunch of things around the cell all at the same time, you stick them all in a vesicle, and then you kind of push that vesicle out there. And what happens actually is that the vesicle gets grabbed by what's called a motor protein that kind of picks it up, grabs it, and then will walk down a highway in the cell, taking the vesicle with it until where it needs to go. And you think that I'm being metaphorical? I'm not. We're going to see a video about this pretty soon, and it looks exactly like that. There's this little molecule, looks like a guy with legs, sort of, that grabs hold of it, and it walks like this down the cytoskeleton. And we've got, like, basically, can't see it in motion because it's too small, but what we have is pictures of the molecule in states, depending upon what type of energy molecule you feed it, and we have, like, pictures of, this foot free, this foot forward, this foot down, this foot free, and they just walk along, carrying their payload until it gets where it needs to go. Okay, mitochondria and chloroplasts. Mitochondria you're going to find in pretty much all eukaryotic cells. Chloroplasts you're only going to find in photosynthetic cells. Mitochondria are the energy powerhouses of the cell. That's kind of what everyone knows coming through like real intro biology or high school biology or something. We're gonna take that and make that a little bit more. We're gonna complicate that for you, just because that's what we like to do, but not quite yet. Now again, you look at the mitochondria and you see these little squigglies, right? Those are membranes that are all crenulated and folded. What type of process is going on here?
Surface area.
Surface area, right. The reaction that generates energy in the cell occurs on the surface of the inner membrane of the mitochondria. So that is, the mitochondria has two membranes, the outer one, which is mostly protective, and the inner one, which is all wrinkly, and that's where the action takes place. Now, not all of the energy reactions in the cell occur in the mitochondria, but the really big ones that generate lots of energy do. Cellular respiration, which is the process that uses oxygen, occurs inside of there. The mitochondria is primarily involved in the production of ATP. So this is where, for the most part, sugar, actually pyruvate, is broken down. The electrons are removed from it. Those electrons are then used to run what is basically an electric motor. And that motor churns out ATP, which is the standard energy currency of the cell. The chloroplast also contains these folded membranes. In this case, instead of being folded like that, these are folded into these what are called granulums, which again look sort of like stacks of pancakes. And in here, the process that's going on is is photosynthesis. And the molecule that actually gathers the light, that receives the light, and transforms that into electrical energy is a molecule that lives on this surface, which is why you've got to have as much surface area as possible in there so you can absorb as much light as possible. The molecules that do photosynthesis and the molecules that do cellular respiration are actually, they're related, they're similar to each other, but they go in opposite directions. Because what you're doing here is not actually making energy, you're not making ATP directly from the sunlight, exactly. What you're doing is you're taking the energy from the sunlight and you're converting it into sugar. You're grabbing CO2 from the atmosphere and you're using that CO2 to build sugar molecules. Then you export the sugar molecules to the mitochondria where they get broken down. Both of these organelles contain an interesting feature. A very, at least to my mind, an extremely interesting feature. Our cells are all controlled by the DNA that's made in the nucleus. These cells, the mitochondria and the chloroplast, both have their own DNA. So the mitochondria has its own DNA. Many of you might have heard of mitochondrial DNA. And that DNA looks like bacterial DNA. It's got a bacterial gene arrangement. They have their own ribosomes inside of them, and those are bacterial ribosomes. The same with the chloroplast. The chloroplast has its own chloroplast DNA, which is bacterial DNA, as far as we can tell. And it has its own ribosomes, and they're very similar to bacterial ribosomes. This has led to what's called the endosymbiotic theory, which is that when, like, the earliest eukaryotes were evolving, probably like the nucleus evolved first. So you maybe had these very primitive cells, and you can actually still find a few of them that, there are a few cells that we know of that don't have either of these. There's lots of things that don't have a chloroplast. You don't have a chloroplast. But there are even eukaryotic cells that we find that don't have mitochondria. And they're really weird in the way that they live. They're definitely eukaryotes. They have a nucleus. They have other organelles. They have no mitochondria. And they can only live through anaerobic respiration. The idea is that sometime very early on in eukaryotic life, some eukaryote out there went and ate a bacteria that was very good at producing energy. And instead of digesting it, something went wrong in that process, and it wasn't able to digest it. And that actually gave it an advantage, because the bacteria, and these appear to be related to what are called, I think, purple sulfur bacteria. The bacteria made more energy than it needed. And so if the cell had extra sugar floating around that it was just doing anaerobically, the mitochondria would take that and make more energy than the mitochondria needed, or what was then, I guess, the purple sulfur bacteria. This is what's called endosymbiosis, where one organism adapts to live inside of another organism. Over time, they became completely interdependent upon each other. The mitochondria is no longer capable of living by itself freely. It lost all of its genes to live in the outside environment because it didn't need them. And now it only has its basic genes for making energy, because it's basically been spoon-fed everything else it might possibly need. Similarly, the eukaryotic cells, for the most part, that have mitochondria have gotten so used to using all of this energy that's produced by the mitochondria. If you take the mitochondria out, cells starve very quickly. Any questions about this?
Yeah.
Yep.
You were saying that the mitochondria was in all eukaryotic cells, but chloroplasts were not?
Right. So the mitochondria is in, like, all eukaryotic cells except a very, very few weird ones. Like, well over 99.9%. Chloroplasts are not. They're only in photosynthetic cells. So, plants, algae, that sort of thing.
And those ones that have chloroplasts do not have mitochondria?
No, they have both.
Oh, okay.
Yes.
Gotcha.
So, question, which of the following organelles contain DNA? Nucleus. Who thinks the nucleus contains DNA? Everyone should be raising their hand. This is the easy one. What about the endoplasmic reticulum? Who thinks that contains DNA? The Golgi? Yeah, one person. Mitochondria? Most people. Lysosomes. Lysosome contain DNA? So the nucleus and the mitochondria both contain DNA. Sorry, the Golgi doesn't. The Golgi is shipping and receiving. Doesn't need DNA. It gets lots of different proteins. Oh. Yeah, that's probably true. I'll fix that. Stupid autocorrect. Thought it would fix that. All right. So we covered most of the membrane-bound organelles, but that isn't all that's in there. This picture here is a picture of the cell where, let's see, I'm going to shut this light source down a little bit. I'm going to briefly turn the lights out so that you can see that picture in all its glory. No, that doesn't work. All right. I have to look at it on here. It's actually a really nice and intricate picture. What it is the different components of the cytoskeleton have been labeled. Just like your body's structure is linked to it by your skeleton, these rigid things inside of you, cells have a skeleton as well. And their skeleton is made of 3 different types of fibers. That's what gives structure to the cell. That's what gives shape to the cell. That's what gives resiliency to the cell. And in addition, 2 of the 3 of these are what are used to transport things around the cell. Because things need to get moving inside of the cell. They need to get from point A to point B. And they don't just magically float there. In a lot of videos that I will show you in the future, and that you have seen before, you see that, like a molecule will just float from over here to over there. They don't, it doesn't actually happen like that. They have to have something that directs them. They have to have a pathway that takes them there. Those pathways are made out of the cytoskeleton. The 3 types of cytoskeletal components are microtubules. These are the biggest ones. They go from the center of the cell, close to the nucleus. There are 2 what are called centrioles, or microtubule organizing complexes. And that's the base of it. And they radiate outward from the center. These microtubules are big, and they're very good at resisting pressure. Like you can push them and they don't compress easy and they spring back. But they don't really have any pull strength. If you pull against them, they fall apart. So they're rigidity structures, but they don't have any tensional strength. These are also commonly used as highways in the cell, especially for big things like organelles. Organelles actually move around inside of the cell. The mitochondria can actually crawl along these and get where it needs to go. And in nerve cells, right, because like the longest cell in your body goes from the tip of your toe to about here in the back of your neck. And it has to transport, like, well, a whole bunch of different stuff, specifically neurotransmitters, pretty much all the way down the length of your body. So those big bulk shipments of chemicals down a nerve cell or down any long cell are going to follow these microtubule pathways. Microfilaments. These are the smallest of them. And they are going to be found in like branches, usually near or close to the surface. So not the blue stuff, but some of this green stuff in here is going to be microfilaments. Microfilaments like to push against each other. Microfilaments are made of actin, and so actin is what the filament is made out of. Myosin is the protein that's going to move down them. If I tell you myosin or myo, anyone here had physiology? What's myosin? Right. You find microfilaments in all cells, but you find a whole ton of them in your muscle cells. And these are what allows your muscle cells to expand and contract, is these actin and myosin, like basically all of the filaments will bunch up when they contract, and then when they expand, they'll go out like that. These also are involved in moving a lot of stuff around the cell, and they're involved in changing the shape of the cell. So for the most part, the cell surface shape, whether the cell is flat or round or square, the basic shape of the surface of the cell is determined by the arrangements of the microfilaments. And those are what gives strength and pliancy to the surface of the cell. The third is intermediate filaments. So if these are the biggest, these are the smallest, these are intermediate. It's pretty easy to guess, right? Intermediate filaments are not highways, right? Nothing travels down them, and they don't have any rigidity. They're like a rope, okay? You can pull on them all you want, and they're really strong when you're pulling, but if you push, they just go bleh. These intermediate filaments are very, very close to the outermost surface of the cell. So these blue things that you see out here, I think that those are the intermediate filaments. They're very close to the surface of the cell, sometimes woven in and out of the cell membrane. So external components. Cell walls. We talked about cell walls in bacteria. What are cell walls in bacteria made out of? Peptidoglycan, right. Cell walls in eukaryotes are made out of a bunch of different things, but they're never made out of peptidoglycan. They are found on all eukaryotic cells. Your cells don't have a cell wall. Plant cells have a cell wall made out of cellulose, which is wood. Fungal cells have a cell wall made out of chitin. Chitin is a different polymer of glucose, so it's sort of similar to cellulose, but has very different properties. I know it looks like it should be pronounced chitin. It's pronounced chitin. Chitin is found naturally in sort of two places, mainly. You find it in the cell walls of fungi, and you find it in the carapaces of insects. So if you want to know what chitin feels like, think of a bug that you squish. That kind of cracky, thin sort of thing, that's chitin. Algae can have cell walls made of all sorts of things. Some algae have cell walls made out of glass. They're beautiful. Some algae have cell walls made out of cellulose. Some algae have cell walls made out of agar. If you've ever seen like on like Discovery Channel or Planet Earth or whatnot, there's giant underwater kelp forests. Those all have cell walls made out of agar or agarose, which is a glucose polymer, a sugar polymer. That's most of what has cell walls in the eukaryotic kingdom, or the eukaryotic domain. Now, moving around. There are a few different ways that things move around in the microbial world. We talked about bacterial flagella, right? You remember the run and tumble? And they have this long whip-like structure that spins around in a circle like a propeller. Eukaryotes have flagella as well. They're totally different flagella. And they work in a completely different way, and they're made out of totally different proteins. Actually, flagella in eukaryotes are made out of microtubules, one of those cytoskeletal components I was telling you about. And instead of spinning in a circle like this, what they do is, it's like they contain two bundles of fibers that like one will push this way, and then the other one will put that way, and then the other one push this way, then the other one push that way. And basically what that causes is that you have a tail that undulates back and forth like this. And that's how they produce their motions. It's a back and forth motion, not a spinning motion. You have two structures which are actually physiologically almost identical except for length, but they're used kind of differently. You can have flagella, which will usually have one or two or three, maybe four max, long tails going back and forth to propel you through. Or you can have cilia, in which case you will have hundreds and thousands of tiny whips that look like little hairs that are all going to beat in unison, and like they're all going to go at the same time and push you through that way. The third way that things move around on the eukaryotic side of things is pseudopodia. The pseudopodia, you see it in amoebas. Most of us are sort of familiar with it. This is like you grow, so pseudopod means false foot. You like grow the false foot, like this big oozy thing out around that way. And it's like you ooze there, and then you just sort of like ooze along that pathway till your entire body is here. And then you extend another little tentacle out, and then you'll just ooze down your tentacle until you show up here. If you want to like eat something, you'll grow a tentacle around it this way and that way. And then eventually, if it doesn't run away, you're just now around it. And that's how you've eaten it. Pseudopodia is usually a much slower way of moving around, but it gives you a lot more freedom of motion. And this is controlled, again, it relies on the cytoskeleton, but you remember I said that what controls the shape of the surface of the cell is the microfilaments rather than the microtubules. So the growth of pseudopodia is dependent upon microfilaments. We have any questions over what we've covered here? Okay. All right, let's continue on our discussion with microbial metabolism. I want to cover a little bit about energy and thermodynamics in the time that we have left. This is mostly foundational material. This is stuff that you kind of need to know a little bit about what's going on to understand how enzymes work and how energy flows throughout the cell. So the first question is, what is metabolism? Okay? And metabolism, we usually think of metabolism in our everyday life. This is one of those words that's different in a scientific context than it is in like an everyday context. Because when we think of metabolism in the everyday world, what do we think of? Digestion. That's actually only half of what metabolism actually is, scientifically. Metabolism is the sum total of all chemical reactions that happen in an organism. Not just breaking things down, but also building things up. Those are the two halves of metabolism. And they have names. Anabolism, which is building things. You're taking basic building blocks and then you're using energy to turn those building blocks into large complex structures. And catabolism. Catabolism is taking big complex things like sugar or proteins or whatever and breaking them down and extracting energy in the process. Both of these processes are a part of metabolism, and both of them are absolutely essential for life. So here we have a cell, okay, undergoing both catabolism and anabolism. Nutrients come in, sugar or amino acids or proteins or whatever. It undergoes catabolic processes. It takes those nutrients apart. It extracts energy from them, and it ends up with fundamental building blocks. It's sort of like Legos. If anyone ever used to play with Legos when you were a kid, you can get like some Lego cars, Lego trucks, Lego trains, and you can take them apart into your little Lego pieces, and then you can rearrange them to build Lego castles or Lego spaceships or whatever. Once you get them into their basic Lego structure, you can rearrange them in whatever way you want. So those are the two things you get out of catabolism, is energy and precursor molecules, which are your building blocks. You then take the energy that you just got out of your catabolism and the precursor molecules that you just got out, your little Lego blocks, and you use your energy to build up whatever new thing that you wanted. Maybe the nutrients that you got were like cars and planes, and you want to build castles and spaceships, because that's what you need to grow. So you use your anabolic processes to build larger, more complex macromolecules like proteins and lipids, and then those go off and do all of the things that the cell needs to do. They make cellular structures like membranes, ribosomes. They control cellular processes like growth and division. Or you can build macromolecules, which are basically ways of storing energy, and precursor molecules that can then go back into catabolism whenever your nutrients run out. So, which of the following processes are catabolic or anabolic? Replication of DNA, what do you guys think? Catabolic? Anabolic? Right. What about the breakdown of sugar in the mitochondria? Catabolic? Anabolic? It's catabolic. Synthesis of new proteins at the endoplasmic reticulum. Catabolic? Anabolic? Good. Digestion of food particles in the lysosome. Catabolic. Anabolic? Anyone? What about building a cell wall? Catabolic? Anabolic? Great, you're all getting the hang of this. You should definitely be able to classify cellular processes as either catabolic or anabolic for the quiz. The cell has a bunch of different energy molecules. We usually talk about ATP as being the primary energy molecule of the cell, and it is. But there are a bunch of other energy molecules as well. You can have glucose up here at the top. That's usually how you're going to store energy long term. It's not readily available, but it's a good way to store energy for the future. You have ATP, which is generic chemical energy. It's good for doing almost anything in the cell, except a few things. It doesn't perform redox reactions very well. That's reduction and oxidation reactions. So it's not so good at building new or breaking down old bonds. It can pair them up, like break one and form one at the same time, but it's very difficult for you to get like a net increase in the number of chemical bonds using just ATP. NADH, NADPH, and FADH2 are all similar. You can see any of these. All of these are specialized energy molecules of the cell. They specifically carry electrical energy or reduction potential. Redox reactions, reduction-oxidation reactions, are actions where electrons move, where electrical energy is used. And those are the molecules for storing and transporting that electrical energy. Let's take a look at an oxidation and reduction reaction in motion. So oxidation means removing electrons from a chemical, and it will usually actually turn a single bond into a double bond. So here we see two carbons bonded together and with their electrons around them, and what we're going to do is NAD plus is going to remove two electrons from that molecule, and it becomes NADH. That's the energy-rich form. NAD plus is the energy-poor form. Now that poor little carbon molecule on the right has an electron imbalance. It only has 6 electrons, it wants 8. So 2 of the ones from the one on the left will come down and form a second bond, and now you have 2 double bonds, and both of the carbons have 8 electrons in their outer shell. That's oxidation. When you take electrons away from something, you are oxidizing it. This was oxidized. The carbons were oxidized. Oxidation and reduction always happen in a pair. If something is oxidized, something has to be reduced. What was reduced in this case? The NAD plus was reduced. In this case, so the NAD plus was reduced. That makes it the oxidizing agent. I know it gets a little bit complicated there, but it's what caused that to oxidize. Reduction means adding electrons to a chemical. This is usually going to break a chemical bond of some sort or another. Might turn a double into a single, or it might cause a single to go away. So the electrons come back out of that bond goes away, and boom, you've reduced your carbon molecules, you've oxidized your NADH back into NAD+. Any questions about that? Good, we're all in review terms for most of you here, I think. So when NAD+ is reduced, what? Electrons are taken from the target molecule. Is that right? Anyone think so? Electrons are donated to the target molecule. Anyone think that is? ATP is consumed. ATP is generated. Obviously, we're not quite going on here. Let me explain what I mean by this. So if NADH is reduced, what is happening to NADH? It's gaining electrons. So the target molecule, the molecule it's acting on, does it lose or gain electrons? Right, so electrons are taken from the target molecule. There are 2 laws of there are 3 laws of thermodynamics, but we're going to talk about 2 of them here. Basically, the first law of thermodynamics states that energy can neither be created nor destroyed, but it can be transformed. So when we say energy is used by the cell, that energy doesn't go away. It doesn't like stop existing. What we mean is that that energy has been transformed from one type of energy that the cell can use to another type of energy that the cell can't use so well. The second law of thermodynamics, there are lots of different ways of stating this. This is sort of the one that I like, is that in any closed system, any process in which energy is transformed, so you're turning energy from one type of energy into another type of energy, this results in a net increase in the entropy of the system. This is sometimes shortened to entropy always increases. But entropy is a kind of difficult concept. It means chaos and disorder, but not in exactly the way we think of it. So like the classic example is a clean room has low entropy and a dirty room has high entropy. That's not actually the case. Both of them have the exact same amount of entropy because both of them have the exact same amount of stuff. If I have two carbon molecules floating in a box, they're both gases for some reason, because carbon is never a gas, but whatever. Let's say that they're gases. This system has high entropy because of the many different positions that these carbons could be relative to one another. They could be far away, they could be close. This one could be on top, it could be on bottom. They could be like this, like that, like this. Anywhere relative to one another. There are a lot of different configurations that could adapt. If I take them and bond them to one another, now they only have one fixed position relative to one another. There are fewer configurations that this system is capable of adopting. The entropy has gone down. We've gone from two particles to one particles. That's entropy in a nutshell. So chemical reactions can be spontaneous or non-spontaneous. Spontaneous reactions are exergonic, which means energy releasing, and they can occur by themselves. So like this ball rolling down that hill, that's a spontaneous reaction. The ball doesn't need any help to roll down the hill. Set it at the top of the hill, it rolls down. And it releases energy. It turns potential energy at the top of the hill into kinetic energy at the bottom. Spontaneous reaction. In this reaction, the delta G, G means energy, delta means change. The change in energy is negative. Your G was high up here. Your G is low down here because you've released and used that energy. So the delta G, the change in energy, is negative. Anytime delta G is negative, the reaction is spontaneous. Non-spontaneous reactions are endergonic, which means energy consuming, and they will never occur unless energy is put into the system. No matter how long you wait, that ball will never roll up the hill on its own. If you put energy into the system, you can force the ball up the hill. We're going to strap a little rocket pack to it. Boom! Blows up the hill, right? In this case, the delta G is positive. This is a non-spontaneous reaction. It needed your help to go off. Unfortunately, that isn't actually the way most reactions look. This is the way most reactions look. This is a spontaneous reaction. You know that it's a spontaneous reaction because if you go from here to here, the delta G, the change in G, is going to be negative. So this is a spontaneous reaction. How long will you have to wait for this spontaneous reaction to take place? Pretty much forever. It's a spontaneous reaction, but that doesn't necessarily mean that it will happen spontaneously. It's kind of weird. It has to do with the shape of this thing. So you have the delta G, which is the net from start to finish. you also have what's called the delta G double dagger. That's called the activation energy. If you put in a little bit of energy to get this to the top of the hill, it'll then roll all the way down to the bottom of the hill by itself. So, I want you guys to think of wood. All right? Does wood burn? Yes. Is that an exergonic or endergonic reaction? Exergonic, right? So it should be spontaneous, right? That's wood. Why isn't it burning? It would be spontaneous for it to burn, right? We just said that that was a spontaneous reaction. And it would be spontaneous, except it needs that activation energy. If I was to take a lighter, light it on fire, I wouldn't have to keep lighting it on fire in order to keep getting it to burn. It would burn by itself once I lit it on fire. That's basically what we're describing here. And now, to get to the biology aspect in like the last minute of class. What do enzymes do inside of the cell? Inside the cell, you have these reactions going on all the time. And what enzymes are going to do is lower that activation energy. They don't change the net energy of a reaction. They just lower that hill and allow the reaction to happen spontaneously. They don't provide the activation energy. They change the shape of the energy curve to lower the activation energy. I'm going to skip by this since we're sort of short on time, but a spontaneous reaction will always have a negative delta G.
Is that a catalyst?
Enzymes are catalysts. Not all catalysts are enzymes. Okay? So these are the important things that I want you guys to definitely know about enzymes. Enzymes are not used up during a reaction. This is also a property of catalysts. The enzyme is the same at the end as it is at the beginning. Unchanged. Enzymes work both ways. All enzymes, if they catalyze a reaction going this way, will catalyze the same reaction going the other way. And what determines which way the reaction goes are the energetics. Which way the energy flow is favorable. Enzymes do not change the net energy. They do not make exergonic reactions endergonic. They do not make endergonic reactions exergonic. They do change the activation energy, the delta G double dagger. Enzymes cannot make impossible reactions happen. If the reaction is impossible, doesn't matter how good an enzyme you have, it's still not going to happen. Enzymes do not add energy to a reaction. There are some enzymes that can take two reactions and couple them together and make them one reaction. And that will change the energetics of it. But the enzyme itself does not add energy to the reaction. And last, enzymes don't always work alone. They often require help from coenzymes, ions, and cofactors. Anyone have any questions? Okay, then I think we're good.